Talking about Staph aureus, Staph aureus is very different from the other Staphylococcus species. Now, when you have a gram-positive bacteria, whether it's catalyst positive or negative, when it's positive, you have a Staph species, and when it's negative, you have a Strep species. Now, with the three important clinically significant ones to humans, there is a distinction between them two, and that is the coagulase enzyme. Now, Staph aureus is able to produce a coagulase enzyme, which actually converts fibrinogen into fibrin, um, and it is seen as clumps in plasma, and that's how the test is done. And Staphylococcus species, actually, specifically Staph aureus, does this to kind of hide itself and be in one place and be localized, and that's how come Staph aureus produces localized infections. Uh, Now, mannitol salt agar, or agar, whatever you call it, is used to grow Staphylococcus species. Now, note that mannitol salt agar allows only Staphylococcus species to grow on it, and so if you culture your inoculate or whatever on it, and then there is growth, then it means that there is a Staphylococcus species present. Now, the original color is pink. Now, when there is growth and then the color changes from pink to orange, then it is obvious that the stuff is aureus. Obvious that is aureus. All right. So the reason is Staph aureus is able to ferment mannitol, and then it changes its color from pink to orange. So Staph aureus ferments mannitol and also hemolyzes red blood cells. So it can cause hemolysis on blood agar. Now, an agar is just a medium we use to grow bacteria outside of the body. Right. Now, you'll notice that the colonies of Staphylococcus aureus here look golden, right? And then it's actually due to a pigment they produce that is called staphyloxanthin, and it's responsible for the golden color of its colonies. The other staph species, albus and saprophyticus, are usually kind of whitish. They have whitish colonies, white or gray. Yeah. So now let's talk about the virulence factors of Staphylococcus aureus. Now, when we say virulence factors, they are characteristics of a microbe or a bacteria that allows it to cause diseases or harm the host. Usually, they help it to evade the host's immune system or defense, and help it to cause its damage. Like the catalyst we talked about, it helps it to escape the oxygen reactive oxygen species mediated killing of the microbe, and then enhance its stay in the organism and causing damage. So the virulence factors of Staph aureus can be divided into three main classes. We have the structural components, we have the toxins, and then we have the enzymes. So when we look here, we can see some of the virulence factors, the toxins, the structural components like protein A, and then the capsule. So we're going to take the structural components first. Now, there is what we call protein A. Protein A. Now, protein A is a surface adhesion and protein, that binds to the Fc portion of IgG. Now, IgG is an antibody which actually binds to bacteria, and that's what we call opsonization. That is, makes the bacteria let me say, sweet, so that phagocytes can, engulf it, right? But then the phagocytes are supposed to bind to the Fc region. This is how an antibody looks like. So this is IgG. Now this is the Fc region, and then this is the variable region. Now, usually the antigen binds to the variable region. This is supposed to have a complementary shape, sorry. And then the phagocytic cell binds to the Fc region to help it engulf the bacteria. But then over here we see that protein A is binding to the Fc portion of the IgG, so it hinders the function of the IgG, that is the opsonization, and prevents phagocytosis and then antibody antigen mediated clearance of the staph species. So virulence factors are actually features of a microbe or a bacteria that helps it to escape killing. Let's simply put. So protein A on the capsule binds to IgG, prevents antibody antigen mediated clearance of the staph. So actually prevents the opsonization. Then we have what we call penicillin binding protein. Now, simply put, before the peptidoglycan layer of bacteria cross-links to give it, let me demonstrate it here. So for example, let's say this is the peptidoglycan layer of the bacteria, and it looks like this. Now they have to cross-link before the bacteria gains some structural integrity that helps it to escape or make it a bit resistant to antibiotics. This is not scripted. So now what happens is penicillin binding protein actually comes to bind to the bacteria this way, and then causes the cross-linking of the peptidoglycan layer, thereby giving the bacteria some kind of structural integrity, and which prevents antibiotics from killing it. So what Staph aureus does is that it kind of changes the penicillin binding protein. Now what we actually do is there's a drug called methicillin, and it works by preventing the penicillin binding protein from cross-linking the peptidoglycan, so that the bacteria will be vulnerable to antibiotics. But then Staph aureus is able to change the penicillin binding protein, and so when methicillin comes here, it is unable to recognize the penicillin binding protein. And then if you can't prevent the penicillin binding protein from performing its function, definitely it's going to cross-link its peptidoglycan and then gain some structural integrity, which will make it difficult for drugs like antibiotics to kill it. So that is one way staph evades destruction. And another one is the polysaccharide capsule. Now Staph aureus has a polysaccharide capsule around it, which prevents phagocytosis. Now when they get phagocytosed, they form a phagosome, sorry, the lysosome, and then enzymes break it down, whatever. If you are not being phagocytized, then it means you are not going to get killed. Now the bacteria stays longer and then causes damage. And teichoic acid on the surface to prevents or facilitates adherence of the Staphylococcus species to mucosal cells